Hey, hello and welcome back again to another video tutorial of Roy's Academy. In this video tutorial, I am going to discuss about a very important experiment that was performed in the early 20th century by scientist Sir Frederick Griffith. Okay, in 1928, in the early 20th century, in order to understand that how there were some implications happening or innate flow of the genetic characteristic or the functional characteristics of a species was happening from one parent generation to the progeny or the offspring. Okay, like if as we know in a normal course of biology, in the alternation of generation, we know that the heredity is the term, that the inheritable characteristic, the phenomena where the specific genetic functions or the characteristic features are being inherited from one generation to another, thereby the progeny generation is also repeating the same thing. Now in this particular aspect further, there were some experimentations that were being performed, like in early in very early stages during the course of development of the biological phenomena and all, during the when the laws of the heredity and the genetics were put forward by Sir Gregor Johann Mendel. He performed that experimentation on the pea plant, where isolated pea pods can be grown in a short course of time. Okay, and the pea pods were clearly distinguishable, the male female characteristics and all. So it was very much easier for Sir Gregor John Mendel in order to identify the experimental procedures and setup, and in order to theorize the distinct characteristics and also develop the theory in course of his observations and experiment. But in empirically, what he did was empirically true. Okay, empirically true means he observed some experiment, and based on some conclusions or observations, he finally came to an perspective, or finally came to an inference. But in the early 20th century, in the course of modern development of the modern biology and all, the speculations needed to be have non-speculated, or the speculations needed to be changed or modified in order to have a firm belief that there was some flow of the genetic material or the heredity from one particular generation to another via some innate medium. There were some soul processes happening within the body of a particular organism during the course of reproduction or mating. Now this is all about how the genetic material was carrying the information from it. So how is the genetic material able to carry the information? What is the capability of that particular genetic material, that it can incorporate the informations or the inheritable traits or characteristic features or specific functions, and can transmit that from one generation to another. Let us see what Sir Frederick Griffith performed in 1928 in the early 20th century through his course of experiment and observed. So the name of the experiment is the test for genetic material by Sir Griffith in 1928. Okay, now what he did, what Sir Frederick Griffith did, he performed a very modified, a very smart kind of experiment through a very easy process. Okay, what he did, he isolated two particular strains of pneumonia causing bacteria. Okay, the first one was R2 type, which was non-virulent, and the second one was S3 type, which was virulent. Okay, the species of that bacteria was Streptococcus or Diplococcus pneumonia. Okay. This was the strain of the bacteria which he isolated from nature. Okay. Now, in his course of experiment, he isolated these two strains from nature. The first one was virulent, that is pneumonia causing. The first one was non-virulent, sorry, non-pneumonia causing, or pneumonia which was not able to produce pneumonia or cause pneumonia. The second one was S3 type virulent, which was pneumonia causing. Now, what he did, he took these two strains of bacteria and took 4 case studies. Okay, let the first be case 1, the second be case 2, the third be case 3, and the fourth be case 4. Okay, he took 4 case studies. Now, as I said, he isolated these two strains of bacteria. Sir Frederick Griffith performed a very smart kind of experiment. Okay, in the era of modern biology, or the revolution of modern biology, he did a very smart way and straight-cut method of experiment. He isolated the non-virulent strain and the virulent strain. The second one was S3 type, and the first one was R2 type. Okay, which was not able to cause pneumonia, and the second one, S3 type, was virulent, which was pneumonia causing. Okay, and the first one was not pneumonia causing. Now, what he did, he cultured these two bacteria separately, and in the case 1, he injected this, he took first, going into the injection phenomena, what he did, he took these two strains in different categories or different styles. Okay, this is the case 1 and this is case 2. Case 2, and this one is case 3. And this one is case 4. Okay, so in these 4 cases, he took 4 different types of 4 different culture medium of these two bacteria. In the first one, what he did, in the case 1, he took only the R2 type living R2 type bacteria. Okay. In the second one, case 2, he took living S3 type bacteria, and in the third case, what he did, he heat killed the S3 type bacteria he took, that is, he killed the S3 type pneumonia causing virulent strain with heat and reduced the virulence, or killed the bacteria or attenuated it. And in the fourth case, he took living R2 plus heat killed S3. Okay, this is what he did. Now, what happened? In the first case, when he injected this culture of R2 type bacteria in a mice, okay, he took some mice, colony of mice, in order to identify the characteristic strain of the of this bacteria. Okay, that how the bacteria was able to cause or perform the disease. So in that way, he came to the conclusion that what was actually necessary for causing that disease, which particular part of the cell, bacteria prokaryotic cell, so which particular part of the cell was necessary for causing the disease. He was hunting for that. In hunt for that particular material or the element which was responsible for incorporating that particular function, he performed this experiment in a very smart manner. Okay, so Frederick Griffith, in what he did, he injected the R2 type living strain within the mice, and as a result of this experiment, what happened? When he injected the R2 type living strain, which was not pneumonia causing, the mice survived. Okay, and in the second case also, what he did, he injected the S3 type culture of the bacteria in mice in the same way like he did in the case 1. Okay, and as a consequence of this, the S3 type was virulent, that is pneumonia causing, so the mice died. Because it caused, this S3 type virulent bacteria caused pneumonia in mice, and it killed the mice. The mice couldn't survive. And in the third case, he injected the heat killed S3, that is, he attenuated or reduced the virulence. He just attenuated means reduced the virulence of the S3 type bacteria so that it cannot cause that particular disease. And similarly, he injected that into the mice. And as a consequence of this, the mouse was able to survive. Okay, because it did not cause the disease, so the mice was easily able to survive with the bacteria inside. Okay, inside their body with the colony of the bacteria. Now, in the fourth culture medium, he took the sample of the living R2 type plus heat killed S3 type. In this case, what he found was very strange and conclusive of the entire fact that of his experiment, the experiment he performed. In this case, he found the mice died. Okay, so this is what the entire setup of his smart and straight cut experiment which he performed in 1928. Now, why in the fourth case, even after heat killing the bacteria, even after heat killing the S3 type, that is virulent bacteria or the disease causing bacteria, the mice died. What was the reason behind that? Sir Frederick Griffith concluded that in the first case, when the R2 type bacteria was given, which was non-virulent, not pneumonia causing, the mice survived because pneumonia didn't happen. In the second case, S3 type or the virulent pneumonia causing bacteria was injected and the mice died. In the third case, heat killed S3 type, that is the virulence or the disease causing effect, the capability of the disease causing for this S3 type bacteria was being totally demolished. Okay, it was totally being diminished from the bacteria, and after injecting that thing within the mice, it was able to survive. And in the fourth case, the living R2 type plus heat killed S3 type bacteria was being given. In that case also the mice died. Now in this particular case, fourth case, what happened? The R2 type living bacteria was present in the culture medium, and along with that culture colony, the heat killed S3 type was present, but still the mice died. So Sir Frederick Griffith concluded that there was some implications or some innateness, some soul causative process which was happening behind the screen or behind the desk, which was going on at the background, which led this living R2 type non-virulent bacteria or non-pneumonia causing bacteria to change into the S3 type bacteria. Why? Because the S3 was heat killed, it was not having the capability to cause the disease or death to the mice, but the R2 type was, in spite of having its non-pneumonia causing capability or non-pneumonia causing effect, the mice died finally. So Sir Frederick Griffith came to the conclusion ultimately that the R2 type living bacteria, which was non-virulent, in the presence of heat killed S3 type bacteria, somehow incorporated the genetic information or somehow incorporated that specific functionality from the S3 type bacteria within its body, and itself got converted to the pneumonia causing S3 type bacteria. The R2 type bacteria incorporated that. So how did the bacteria incorporated that? There was some hereditary characteristic or hereditary trait that the S3 type bacteria was having, which rather when entered into the body of R2 type bacteria, it changed or modified or metamorphosed the hereditary characteristics of the R2 type bacteria. Hence the R2 type strain converted into the S3 strain, and finally pneumonia was caused, and pneumonia happened within the body of the mice and the mice died. As simple as that. So this is how Sir Frederick Griffith, in the early 20th century, he was the first and foremost scientist, or one of those major scientists who shook the entire world of modern biology or cell biology in course of its evolution, or brought about a revolution in order to depict the phenomena, or in order to illustrate, validate the phenomena that there was some innateness that was happening in the background scene or background process that led the conversion of the heat killed S3 type to the R2 type, and that was the only material, that innate material or the sole causative process or the root cause was the only cause which helped in leading to the functionalities of a particular organism, or the characteristic feature of a particular organism. Either it's a bacteria, either it's a multicellular species, either it's a prokaryotic or eukaryotic organism, whatever living cell it might be. There was some innateness within that particular cell, there was some material within that was present in the cell that led to this phenomena. So that's how Sir Frederick Griffith concluded in his hunt for genetic material in 1928, that how the phenomena happened and why, and he named this particular phenomena as transformation. Okay. He named this particular phenomena as transformation. transformation means nothing but the R2 type strain transformed into the virulent S3 type strain, which helped lead the development of pneumonia, and the mice died finally. Okay, so this was the ultimate conclusion or inference of his experiment, that the phenomena which the DNA was having was transformation. The genetic material was having the transformation. Okay, that DNA or the genetic material is basically nothing but was bound to be some innate process or innate root cause or molecule that was present in the cell which can transform. And he finally led to the conclusion or inferred that DNA was that material which was able to transform easily. So Sir Frederick Griffith concluded that through this transformation principle, it was possible that one type of DNA can incorporate the genetic informations or the nucleotides or the genetic material from the molecule of the other cell, and can itself develop or transform into that particular that particular cell itself, or the have alternate characteristics of that cell by suppressing the innate characteristics of itself. Okay, so this is what all about Sir Frederick Griffith's experiment was, and how it led to the evolution of modern biology, that how the transformation concept changed and brought the limelight of the development of development and understanding of the molecular biology, that DNA was the sole root cause of each and every processes happening within the body. So if you like this video, subscribe my channel, share the video as much as possible, hit the like button, and comment down in the comment section below if there are further more queries or doubts. Thank you.